Okay, so thank you very much for inviting me to talk today about the work we've done. We have been investigating duplicated genes, so paralogs, and kind of the functional fate, so what happens after the duplication in terms of function. And we used expression profiling, and also there's some structural analysis built in at the end. So closer to them. Good? Okay. So the function COSI is a very broad audience, and I want to start by just talking orthology versus paralogy. So here, this is a gene tree, and so we have two copies of like two human genes within the gene tree, two mouse genes, due to the duplication event that happened in the ancestor. And so orthologs are genes, so extant genes that are related through speciation. So this human gene is orthologous, so it has an orthologous pair to each of to this mouse gene, the Drosophila gene, and the yeast gene. And this human gene also has its orthologs. Right. And then paralogs are the genes through duplication. So we can color the gene tree and then say these are groups of genes, and each of these genes, so within the gene set, genes are orthologous, we expect them to be more functionally related. And then between the gene sets, we don't know which one is the functional correspondent. And so, as was previously discussed, and about the ortholog conjecture has been kind of something of much debate, and hopefully not too much continuing debate. But this is kind of related to what we're talking about, but not quite the same thing. So where orthologs are more functionally similar, not equivalent, than paralogs. And so, yes, there's members of the audience, so Predrag and Wyatt, that have previously worked on this. I was going to say generally accepted. We can see.
A blood will be really hard to get out of the carpet.
I don't want to poke the bear too much. And so here, instead of this, I'm going to talk a bit about how we see orthology and trying to work out which one of those orthologs. So when we've got duplication events, the orthology becomes very complicated to work out the orthologs, the correct orthologs, as everyone said. And so this yeast gene doesn't just have one ortholog, it's 1 to 2 in this case with human, or 1 to 2 with mouse, etc. And so when we're attempting to like propagate function knowledge that we know, we kind of want to know which one of these human genes we should go to. So one of the concepts that is useful for this has been developed by back in, I think it's 2010, so quite a while ago, in the Panther database developed by Paul Thomas's group. And this is the concept of least diverged orthologs. So in terms of a gene tree, this looks post-duplication event. So for each of these human genes, we'd be comparing the branch length following duplication, and then the least diverged ortholog in terms of sequence, because the species tree is based on sequence, would then be defined as the least diverged ortholog. And then the assumption that we make based on this is that the least diverged ortholog is predicted to maintain the ancestral gene function. So that's the function, I don't know whether I've got a pointer here in, I also put an arrow, so it's helpful. But in the ancestral gene here, so the shorter branch following the duplication event, the assumption is that this is going to maintain this function. So back 2-3 years ago, Paul visited us in Switzerland for a sabbatical, and we kind of all got together, Natasha, Christoph, and Paul, and Irene, and we kind of thought, no one's really tested this. And so we kind of set out to test this. And similar to the ortholog conjecture, we wanted to define this kind of in some formal-ish kind of way. And so we came up with the notion of the least diverged ortholog conjecture. So that following this gene duplication, we think that the least diverged copy in terms of sequence is more functionally similar to the ancestral gene. Okay. And so what does this mean? If we look not at extant genes, so the human genes, we look at the ancestral genes that are inside the nodes, inside the tree. So the ancestral pre-duplication, gene 1, 2, and 3. Then we can, for every gene tree, we can color this as the least diverged copy, the most diverged copy, and then it's so here in this plot, it's gene 1 and gene 2 that we're predicting are more functionally similar. Of course, we can't have the data for the ancestral states, and so we kind of want to first of all formalize the kind of framework that we want to test this in, and then I'll explain the data that we used to investigate this. So one of the problems with testing this is that on the gene tree, when you're defining the least diverged ortholog, you always can define this as the shortest path. So this is the shortest absolute path. Even if you had kind of one like very small difference, we would still end up classifying one as least diverged, one as most diverged. And so in order to identify, we wanted to split between symmetric evolution and asymmetric evolution. So where you've got clear differences in the branch length following the duplication event, we needed to have some kind of method for assessing each branch's significance from the divergence of the normal kind of evolutionary, the kind of rate of evolution in the gene family. And so this is kind of a bit of a problem because different gene families evolve at different rates, and the gene trees could look like this or this. And we need to kind of normalize these branch lengths so that if we see just an absolute very long branch, this might not necessarily be like significant if the whole gene tree is like super highly divergent. And so the method that we ended up using to like help with this was the looking at like attempting to find a specific like a family specific scaling factor in order to scale each of the gene families to come up with this rate of evolution. So for each given family, we have a correspondence between the species tree branches and the gene family branches. And then we model this basically as that each branch in the gene tree is the branch in the species tree with a small error. Well, it's dependent. And then in the case of losses, we end up saying that the branch in the gene tree is corresponding like the sum of the two branches by the scaling factor. This allows us to kind of build up this massive system of linear equations across all of the gene families from Panther. And then we have the species tree, and then we can end up kind of building. One step is that we fit from the gene trees, we fit the branches in the species tree. And then the next step that we fit back the family specific factors, so how much you scale the gene family. And we kind of keep on doing this until we converge. So we end up then having these factors for each of the gene families to describe how fast they evolve. Then across all of these, so more than 15,500 gene families across over 140 species, we can then perform significant tests on the branches within the gene families to classify whether they are shorter than expected or longer than expected. Because under our model, we can define the expected branch length, and we look at the 5% per branch that are shorter, 5% longer. This allows us to classify into these 6 different categories, whether they're short, normal, long, which then correspond to symmetric and asymmetric evolution. So across the tree of life, we see that the majority of duplication events end up exhibiting symmetric evolution following the duplication event. And then there are many across the tree of life that are asymmetric. The vast majority of the duplication events came under these first two categories. So the normal, where both branches are following a kind of usual evolutionary rate, and then asymmetric, where one of them has shot off and changed. So how do we actually go about testing these functional differences? So previous studies on the ortholog conjecture used gene ontology. We kind of felt that there's some issues possibly with authorship bias when considering experimental data. And so we wanted to use expression data, and just to say, this has previously been used in some other studies, but they tend to be small studies with only a few species. So the assumption that we're using here in this study is that genes that co-express tend to have similar functions. And so therefore we can use the similarity between their expression patterns as a proxy for functional similarity. We have RNA-seq expression data that we collected from the BG database, which is like co-located on our campus in Lausanne, the team that developed this. So there was an overlap of 16 animals that's in their database with the species that we have in Panther, and also Irene had previously worked in the group that developed the plant expression omnibus, and we have 20 plants from this. So after performing like a process to normalize the data, we can end up then having a similarity measure between these profiles. We use Pearson's correlation coefficient. So we decided to perform two different tests. So the first expression test was just, is there a difference between these copies? So following because we don't have the ancestral data, we have the extant data, between these two human genes or between the two mouse genes, we performed, for all the pairs that we have the data, we computed the correlation of the expression profiles. And so we're classifying blue, symmetric, orange, asymmetric. And so for this, we saw, yes, there is a difference. It's slight, that in the asymmetric case, the expression similarity is slightly lower, but this is significant in both the animals and the plants. This is statistically significant, though, change. And so kind of the more interesting test that we did, because the previous one just says, yes, there is a difference in terms of function, is using a co-orthologous outgroup gene. So for example, if we wanted to test the humans, we could use the yeast outgroup, mouse, the same yeast outgroup. And then we concatenate all of these together. We chose the closest outgroup species that we have the data for in order to perform this. So then we also did this. So we always classify one as LDO, one as MDO, and then using the closest outgroup. For this, we had to then have similar anatomical entities. So between the two species, and we filtered out the species where we didn't have enough data, essentially. So this test, we wanted to, rather than just have all of the duplication events in one column, we can take the divergence times from time tree of the speciation events that ended up being studied, where we've got the link to the co-orthologous outgroup. And so across time, so this is in the symmetric case for the least diverged copy, we have this decrease in expression similarity over time, which we also view with the most diverged ortholog. However, it is less functionally, less similar in terms of expression and thus function. The asymmetric case, least diverged, is also, and then for the most diverged ortholog, we see that this is much lower in terms of expression similarity, and we kind of think that these results in both cases, in the symmetric and the asymmetric cases of evolution, these suggest that function of the least diverged ortholog is more conserved. So then, just very briefly, because there's not much time, what about the rest of the duplications? So we know that protein structure is highly related to function, and so AlphaFold has given us this huge database of, so we can download all of the structures for the 143 species. We can then use FoldSeek in order to align these structures that AlphaFold has given us in order to have structural similarity scores. And then using this instead as a proxy for function, instead of the expression, not together, we can also perform the same test that we've just performed with the expression for the co-orthologous outgroup. And so here, instead of expression similarity, the y-axis is the structural similarity. And then you can notice that across the x-axis, the time since speciation, we're managing to span the whole of the species tree that Panther covers. And so this, again, across time, we see that the structural similarity of more recent duplications are higher, and it's lower for the branch that's diverged more. And then in the asymmetric case, we have a similar pattern of structural similarity across time. However, when we look at the most diverged ortholog, so in the asymmetric case, this is significantly, well, it's much lower similarity than the kind of bands that we have across here, where both copies following symmetric evolution are in a similar part of the graph. So just to summarize, we found that the symmetric events exhibit higher structural and expression similarity than the asymmetric, suggesting a change of function. We think that we saw that under the asymmetric evolution, the longer branch has much less structural similarity to the outgroup. And we kind of feel that these results are consistent with our definition of the least diverged ortholog conjecture. So following the gene duplication, the least diverged copy is more functionally similar to the ancestral gene. And thank you very much for listening. And thank you to Christoph, Paul, Natasha, and Irene for their great work in this project. Thank you.
Okay, so there's no any online question. Is there any question in the audience?
So I have two questions. So first, if the LDO has worse sequence of structure similarity than the MDO, which one I should use for function prediction?
The LDO has.
Here your data suggests that LDO is more conserved than MDO. But if LDO has worse similarity to the target sequence than MDO, then which one I should use?
So the closest sequence isn't necessarily.
So the closest sequence in a phylogenetic tree is not necessarily the sequence with greater similarity, right?
No.
So which one should I use? Should I trust the similarity more or the phylogenetic tree more?
I'm not sure.
And also, if I know both the function of LDO and MDO, why shouldn't I use both for function annotation?
Because if we've seen that there is a significant change in the sequence, surely there's been a change in the function.
Okay. Yeah. So I guess at least we should test whether similarity or phylogenetic tree distance has greater correlation with function similarity.
Yes. Yeah.
Thank you.